Hello and welcome to today's webinar where we present the sample multiplexing capabilities of our Tapestri platform. Our speaker for today is Jacqueline Marin, product manager here at Mission Bio. Jackie worked as a research scientist for a decade, mainly in the field of single cell, before and joined Mission Bio 6 years ago. She started in our R&D team and transitioned to product management in 2022. And as we have some time for questions at the end of Jackie's presentation, please type in any questions you might have in the Q&A chat box on the bottom of the screen. And yeah, with that, I would like to hand it over to Jackie.
Okay. Thank you so much for that kind introduction. Hello, everyone. Welcome to our webinar, which we have titled Multiplexing 101, Unlocking Critical Single-Cell Insights with Increased Throughput and Efficiency. Here at Mission Bio, we have a distinct technology. We're the only single-cell platform that enables targeted and high-throughput single-cell DNA sequencing with multi-omic characterization integrating genotype and immunophenotype, and we also offer custom target assay development. Mission Bio at a glance, we're in over 70% of NCI cancer centers with over 97 publications. We currently have 34 catalog panels and growing, and we have sequenced over 10 million cancer genomes. The Tapestri platform seamlessly integrates into existing NGS workflows. You start off with your target selection. You can either have a pre-designed catalog panel, or you can design a custom DNA panel. From there, you're able to utilize our Tapestri platform, our instrument, to do single-cell workflows for DNA and protein. Once you've completed your work on Tapestri, you would then go into sequencing, analysis in our integrated pipeline, and then be able to do further analysis with our tertiary software. How the Tapestri works: first, you would prepare your single-cell suspension, and then you would be able to encapsulate those single cells on the Tapestri, introducing a protease as well as an oil to that sample to create single droplets. Then you would lyse that sample on a thermocycler and reintroduce that emulsion into the Tapestri instrument, into the cartridge once again. And you would include barcoding beads and an appropriate reagent mix in order to amplify your targets of choice and move into your library prep. Once your library is completed, you would sequence it on a Illumina sequencer, and then you'd be able to analyze those FASTQ files using our pipeline as well as our additional visualization software. So here we introduce sample multiplexing. We're lowering the barrier of entry to Tapestri and making single-cell multi-omics more accessible. The benefits of doing multiplexing: it makes single cell more cost-effective, have higher efficiency, higher throughput, and ultimately time savings. Some relevant applications that would benefit from introducing sample multiplexing into their experiments are those working in disease modeling, whether it be working with heme malignancies, with patient-derived xenografts or PDX, or organoids. For those working in gene editing, if you're working in cell and gene therapies or disease modeling, you would benefit from adopting sample multiplexing as well. Our multiplexing software capabilities are now available, and we've introduced two different types of multiplexing. We have antibody hashing and genotype multiplexing. Our antibody hashing solution is available for our standard DNA only and genome editing DNA only pipelines. However, it will be available for our DNA plus protein offerings in the second half of 2024. Our genotype multiplexing is currently available for our standard DNA only and standard DNA plus protein. Delving deeper into sample multiplexing by antibody hashing, we're introducing a core capability to Tapestri that has been universally adopted by other single-cell technologies. We're doing this by staining your samples up front with a hashtag AOC or antibody oligoconjugate. You're then able to pull those samples, run them on Tapestri, so completing encapsulation and barcoding, and then moving into your library prep and sequencing. From there, you'll be able to demultiplex those samples based off those hashtag AOCs using our Tapestri pipeline software, and then do further analysis with our tertiary software. Again, this is available for our standard DNA only and genome editing DNA only currently, and will be available for DNA plus protein in the second half of the year. By adopting antibody hashing, you'll be able to reduce the price per sample by up to 60% and save 1 day of hands-on time. You'll be enabling high throughput processing by multiplexing up to 3 samples at a time. Additionally, you're able to multiplex samples regardless of genetic relation. So this allows for longitudinal or same patient retrospective studies. This is available through the BioLegend TotalCT hashtags and our existing core kits. The changes to the workflow when adopting antibody hashing are as follows. For your cell prep, you will now be staining your samples with a hashtag AOC and combining those samples prior to running on Tapestri. On Tapestri, there are no changes to the protocol. However, in library prep, for those who are doing DNA only currently, they will be adopting our DNA plus protein workflow in order to extract their DNA and protein library, which contains the hashtags. Sequencing parameters do not change. However, in analysis, you will be selecting the demultiplexing samples module, which is titled Antibody Demultiplexing for antibody hashing. By running 3 samples in parallel, you will be saving 1 day of hands-on time. And for those customers who are currently doing DNA only, there will be an adding of 3 hours of hands-on time to stain your samples. Here we have an example of antibody hashing and it effectively separating samples. We have a mix of 3 PBMCs, and here you can see them clearly distinguished by their protein signature. So PBMC1 has only hashtag 1, PBMC2 has hashtag 2, and PBMC3 has hashtag 3. Moving into sample multiplexing by genotyping, this is unique to Mission Bio. We're leveraging genetic information to distinguish your sample. Something very important to note with genotype multiplexing, you're mixing genetically unrelated samples. You're able to take them and pull prior to running on Tapestri and completing your encapsulation and barcoding. Then you'll subsequently move through your library prep and sequencing, and then you will be able to demultiplex those samples based off that genetic information via our Tapestri pipeline software, and then move into further analysis with our tertiary software. This is currently available for our standard DNA only and standard DNA plus protein. By adopting genotype multiplexing, you'll be able to save up to 60% in cost per sample and save roughly 2 days of hands-on time. By multiplexing 3 samples, you're enabling a high throughput processing, as well as you're able to multiplex these samples with minimal cell manipulation. There's very few changes to the workflow, and this is available with our existing Tapestri core kits. The workflow changes when looking at genotype multiplexing are as follows. In cell prep, you simply have to mix your samples before running them on Tapestri. Your Tapestri workflow, as well as your library prep and sequencing, do not change. And during analysis, you will be selecting the demultiplexing module, which is titled Genotype Demultiplexing for genotype multiplexing. By running 3 samples in parallel, you're saving roughly 2 days of work. Here we have an example of 3 samples that were mixed using genotype multiplexing. These are bone marrow mononuclear cells, and here you can see that each of them is distinguished by a particular genetic signature. We use germline SNPs in order to separate the samples. So here we have all of the SNPs that were used here on the heat map to distinguish each sample from one another. Some of the sample multiplexing resources that we have available, we have flyers for each type of multiplexing, as well as an application note for antibody hashing and looking at working with PDX to accelerate drug discovery, and also another looking at our genome editing solution and multiplexing samples within that application. Lastly, we have a poster that is in the CGT space and has multiplexing added to the experiments. And I believe this was presented at ASGCT quite recently. A preview of our application notes. So the first one that I want to highlight, we have named Beyond Basic Characterization, the synergistic power of Tapestri and patient-derived xenograft models to accelerate drug discovery. Our background is that despite the swift evolution of available treatments for AML, a definitive cure remains elusive. And so leveraging the synergy of single-cell DNA sequencing and PDXs has empowered us to unveil clonal dynamics, pinpoint genetic mutations, and delve deeper into the intricate evolutionary trajectories of tumors. For example, developing mechanisms of resistance. The key takeaways for this application note are firstly that we have expanded our capabilities within Tapestri to include antibody-based sample multiplexing, and that with the Tapestri, we're able to discern changes in clonal architecture prior and following treatment with on-market and exploratory compounds. And this has allowed us to shed light on treatment responses, compound efficacy, and disease progression. Our first figure here, we have two heat maps. The heat map on the left, or panel A, are the untreated samples, or untreated PDX AML samples. And on the right, panel B, we have the treated AML PDX samples. What I really want to highlight here is that model 1 and model 2 were actually generated from the same patient. And so this truly highlights the power of antibody hashing being very flexible in the type of samples that you mix. You're still able to distinguish them despite being genetically related. Here, we're highlighting model 2, and this was treated with a compound Y daily for 9 weeks. And you can see that 2 new subclonal populations with additional mutations were detected after 9 weeks of treatment with the treatment schedule with compound Y. And this potentially shows occurrence of mechanisms of resistance to compound Y. Next, we have model 3. And this was treated with venetoclax and a compound Z for 1 month. And so here we see post-treatment, the clonal population with mutations in what we have anonymized as gene 3, which is an indel, and gene 4, which was a SNP, are completely eradicated. And this shows that the treatment with venetoclax and compound Z was very efficacious and has eradicated those mutations. Next, we have our other application note, and this is high throughput assessment of genome edited cells with Tapestri antibody hashing. Our background is that our Mission Bio Tapestri genome editing solution enables high throughput analysis of genomic edits at single cell resolution. Tapestri measures both indels and deletions and single cell nucleotide variants, providing precise data of on and off target edit frequencies, as well as zygosity and co-occurrence. And here we demonstrate how antibody hashing enables high throughput processing of gene edited samples while preserving high reproducibility, sensitivity, and specificity. So the Tapestri genome editing solution, along with antibody hashing, provides very similar performance metrics to a single sample run at a fraction of the price and in less time. So here in figure 1, we show our consistent and reproducible results. These are correlation plots between these 3 runs, or 3 multiplex runs that we did with these gene edited pools. And here you can see we have 3 different pools, one that has a PDCD1 edit, one that has a TRAC edit, and the other that has a combination of PDCD1 and TRAC. And so here you can see a very high correlation between the results that we're seeing in each run, with an R squared of above 0.99 for each run. Next, we have a heat map of the on-target editing co-occurrence using these 2 different guides, TRAC and PDCD1. So the first heat map is the multiplex run. So this pool with PDCD1 and TRAC was ran with the other 2 pools. So the pool that has the single edit in PDCD1 and the other pool that has a single edit in TRAC. The singleplex run, which is on the right, was just ran on Tapestri on its own. So it just has PDCD1 and TRAC. The comparison of the multiplex and single runs are comparable for the dominant clone with biallelic edits of TRAC and PDCD1. And we see a very tight spread between 84 and 87%. Here in figure 3, we see the indel profile guides for the experiments. And so on the left side, we see the expected indel profile for each respective guide And this is as detailed in the Synthego ICE tool. Each guide has its own signature of indels, and they're only present in the pool which they were used. And on the right side, we see the indel profiles of the guides that were used in the CRISPR-edited pools, as shown in our Tapestri genome editing solution. For those that are interested in adopting antibody hashing, all of the components are currently available for purchase. You'll need required reagents, a DNA plus protein core kit, as well as a cartridge kit and your respective DNA panel. The new portion is purchasing those antibody hashtags, which can be done through BioLegend. We currently have 3 available, and the SKUs for each are listed here. The second piece is the software element, and the demultiplexing module is now available on Tapestri. And for antibody hashing, it is available for standard DNA as well as genome editing DNA. For those interested in adopting genotype multiplexing, all of these components are also now available. And you will need a DNA or DNA plus protein core kit, depending on which assay you are doing. You'll also need a cartridge kit as well as a Tapestri DNA panel of your choosing. The new element is our bulk NGS ancillary kit, which we launched last November. And this will provide you the bulk information necessary to distinguish your samples if you do not already have it. To seamlessly integrate that bulk information into your experiments, you can purchase the bulk NGS ancillary kit and use it in your experiments. Then you also have your software piece, and the multiplexing module for genotype multiplexing is now available on our Tapestri pipeline, and it's available for standard DNA as well as standard DNA plus protein. Lastly, we wanted to take the opportunity to highlight some of our upcoming releases, so our expanding applications on Tapestri. Very soon, we'll be having our Tapestri multiple myeloma multi-omics assay launch, and this will allow you to assess SNVs, CNVs, BDJ clonotypes, and surface immunophenotype in myeloma cells in a single assay. It will allow you to uncover comprehensive clonal and subclonal insights into myeloma evolution, druggable surface protein expression, and therapy resistance. We'll also be launching our genome integrity CMV assay very soon, and this will allow for large-scale assessment of copy number variations across the entire genome. You'll be able to uncover the clonal heterogeneity of tumors to understand the clonal evolution and resistance, rapidly assess genome integrity in iPSCs, stem cell banks, and drug products, and ensure absence of CNVs in genome-edited cell therapy. If you'd like more information, please visit missionbio.com.